Today, it's a pleasure for me to talk with a friend of mine, Bindu Paul. She's an assistant professor at Johns Hopkins University School of Medicine, where she's in Department of Pharmacology and Molecular Sciences. She's also has an appointment in the Department of Neurosciences, where she spent many years working with Saul Snyder, who's a very well-known neuroscientist who made a lot of important discoveries, including one we'll talk about today. So the topic for today's podcast is brain gases and their roles in health, normal brain function, and disease. And most people understand that neurotransmitters, as for example, amino acid-derived neurotransmitters such as dopamine, serotonin, or amino acids themselves, glutamate. We've talked a lot about those in previous episodes. But it turns out that neurons in the brain produce gases. And Bindu is going to talk about three gases. One is nitric oxide. Another is really surprising, carbon monoxide, which everybody knows is being toxic. And we hear about reports of people dying in cold weather because they're like burning something that's burning fossil fuels, and carbon monoxide is produced and they essentially suffocate. But it turns out carbon monoxide is produced by cells in the brain. And then finally, hydrogen sulfide, which Bindu has devoted a lot of her efforts to recently. So Bindu, before we start talking about these brain gases, could you tell our viewers and listeners where you grew up and how you got interested in science and neuroscience in particular?
Yeah, sure. First of all, let me thank you for inviting me to share our work with everyone. Yeah, and regarding where I grew up, so I grew up in India. I'm a citizen of India still. And I was basically born in a family where my dad was a naval officer. So I traveled all over the country. So I did not stay in one place. So I was exposed to diverse cultures and climates, and I studied in 7 schools in 10 years. But then all during my time, I was always interested in science. And that was first realized when I joined the Indian Institute of Science in Bangalore, the Silicon Valley of India, for my PhD. But then my thesis topic had nothing to do with what I'm doing now. I started off as a microbiologist, and I worked on a bacteriophage called mu, so which infects bacteria. But the common theme in all of my research was transcriptional regulation. So I was studying the transcriptional regulation in this bacteriophage. And
so how their genes are regulated?
So they have, early genes, middle genes, and late genes. So the transcription proceeds temporarily. So I was working on one of the late gene transactivators, which is responsible for the virus maturation. And I was mostly focused on transcriptional regulation. And this protein that I was working with was a very high affinity DNA binding protein. So I actually ended up exploiting that high affinity binding to develop an artificial regulatory circuit where for stable expression of toxic proteins. So using this protein as a repressor, the E. coli, BL21, BL26D3 cells. So that was kind of.
But Bindu, so you say that work didn't seem to have much to do with your current work in neuroscience, but actually, from an evolutionary perspective, right, these bacteria produce some of these gases. Yes. In fact, hydrogen sulfide, I think.
Yes, that's right.
So, but you didn't really know
Yeah.
about that probably then.
No, I did not. And I did, of course, everybody knows about hydrogen sulfide. It's just an environmental toxin. And you know that the bacteria produce it. And most interestingly, you know that the smell of rotten eggs is due to the presence of hydrogen sulfide. So it used to be infamous because of its
And
bad
also volcanoes. When volcanoes erupt, my understanding is they release a lot of hydrogen sulfide.
Exactly. It produces a lot of hydrogen sulfide. In fact, life evolved in an H2S rich atmosphere. The atmosphere of the Earth, billion years ago, like Earth is 4 billion years old, right? Around that. So in the early atmosphere, you had more hydrogen sulfide than oxygen. So, and the life forms which evolved during that time relied on hydrogen sulfide, especially if you look at the deep sea vents. You have all those hydrothermal vents, so which used to spew out this so-called toxic gas, but then that used to be taken up by the sulfate reducing bacteria and used for as an energy source. So that is how it's actually an ancient signaling molecule, which evolutionarily we have retained so that it can carry out some modern functions, right? So
yeah, and I've been interested in this for years now, that early life evolved in, well, life in general involved in very harsh environments.
Yeah.
And the individuals, organisms that were adapted to be able to tolerate those toxic chemicals, whether it's hydrogen sulfide or even things like iron and selenium, these metals, they're toxic at high concentrations. But organisms evolved not only to be able to tolerate these toxic chemicals, but to use them to their advantage. And I think that's kind of a good perspective on this, that we evolved to be able to use these toxic gases to our benefit.
That's right. So this is also what Paracelsus mentioned, right? The dose is a poison. So yeah, too much oxygen is toxic, too much water is toxic. So yes, hydrogen sulfide, it is beneficial in low doses, but at high doses, it can be really, really toxic. In fact, that is how, the effects of hydrogen sulfide on the human body was discovered. Because in the 17th century, in cesspits and, sewage, there used to be a lot of H2S being emanated, and the workers working there to clear that area would get eye irritation. This is in the 17th century. And also they had some copper coins in their pockets, which would turn black because of the hydrogen, but they did not know what it was that turned them black. So
that's interesting.
Yeah.
And also, carbon monoxide, obviously toxic in high amounts. And that was probably discovered back in like caveman times when they started fires in a cave and then essentially, suffocated and learned that they can't be having fires in an enclosed. Of course, they didn't know anything why back then, but that's why.
That's right.
Okay, so then how did you end up in Baltimore at Johns Hopkins?
Right. So, yeah, there is a consensus sequence right here. So I studied in Bangalore and then I came to Baltimore. So be more that way. Yeah. So but I did not directly come to Baltimore. I first did a postdoctoral fellowship with Dr. Yun Boshi at the National Institute of Child Health and Human Development at the NIH in Bethesda. So I did a 4-year postdoctoral training with him. And here, once again, I worked on a totally different field. This was developmental biology, but then I used all the principles of the prokaryotic transcriptional activators and repressors in this field. So I ended up studying the molecular mechanisms governing thyroid hormone action in frogs. So Yes, so I studied thyroid hormone receptors, and I developed dominant negative mutants of some of the co-activators and showed that they were essential for metamorphosis. So that's, and after that.
So the thyroid hormone is very important in a tadpole that swims,
yes.
then changing or metamorphosing into a frog.
Yes.
Yeah.
So yeah, it's very interesting. So if you just take tadpoles and just put them in a tub of water and you add thyroid hormone into it, they'll transform. So that's a single hormone can induce that change. So that is what is fascinating about the system.
Yeah.
And the development is all external, so you can study that. So, and after the postdoctoral fellowship at the NIH, I came to a turning point in my career in the US. At that time, I was on a J visa and I needed to change my immigration status. And so at that time, NIH wasn't supporting H visas.
No.
There was some kind of a freeze. So I started looking at other labs, but all the while I knew that Saul Snyder's lab was the lab that I needed to be, because even as a graduate student, during journal club, I would present the papers from his lab, especially the ones on Huntington's disease and some other neurodegenerative diseases. At that time, I just knew that Saul Snyder was a very famous scientist, and he did stellar work in the field of neurotransmitters and in neurodegenerative diseases. But I had no idea that I would be joining this very lab and working on the exact same problems. So I had read his paper in Nature on Huntington disease together, which was also authored by Chris Ross, and I was fascinated by the field. So I thought, okay, why not look at transcriptional regulation in Huntington's disease or Alzheimer's disease? So I sent an email to Saul at 8 a.m. in the morning. And then immediately within 10 minutes, I get a reply from Saul saying, oh, let's meet and chat. So this was in December of 2004. And Saul invited me to his lab. That was just the day after Christmas. I remember that. And so the university was partially shut down because everybody was on vacation. So it was just Saul and me. And then we went out for lunch in the Hopkins cafeteria. And Saul asked me a series of questions, but mostly about family, because Saul always puts people first. And then he asked me about my science. So we had a really good conversation. And then we decided that his lab would be a great place to explore all my idea. So that's how in 2005, I joined Saul's lab and started working on redox active molecules like hydrogen sulfide and ergothioneine, which is one of the small molecules I studied. And then the rest is history. So I've been there ever since.
All right. And Saul, David Brett in his lab had cloned an enzyme. It's called nitric oxide synthase. It's an enzyme that essentially produces nitric oxide. Nitric oxide is a gas. And that was really fascinating. And then they looked in the brain at where that enzyme was. And it seemed apparently not to be in every neuron. It's in a small subset, apparently, at least at high levels, of this nitric oxide producing enzyme. So that suggests that somehow those neurons, nitric oxide in those neurons may be having effects on other neurons. And can you talk about, historically though, nitric oxide as a signaling molecule was discovered before Saul started working on it by people who are studying blood vessels.
That's right.
Can you like briefly summarize that work, which led to a Nobel Prize?
Yeah, so basically nitric oxide was first recognized as an endothelial derived relaxation factor. So the Nobel Prize around that time was awarded to 3 people like Louis Ignarov, Furchgott, and Farid Murad there were 3 laboratories simultaneously working on nitric oxide, and the chemical identity of the endothelial derived relaxation factor, which relaxes blood vessel, it was a factor at that time, was attributed to nitric oxide by Louis Ignarro. And so basically they found that, and subsequently it was found that it activates the guanyl cyclase, which produces cyclic GMP, which is a second messenger molecule, which is responsible for relaxing blood vessels. Yeah. So this is what, this was the first instance of like a gas acting in through a signal transduction pathway. And of course, we all know now that the first enzyme which generates, which was cloned, which generates nitric oxide, was the neuronal nitric oxide synthase, which was isolated and cloned by David Brett and Saul Snyder. And it so happened that this regulates a lot of the signaling pathways in the brain, and it is a very, very important neurotransmitter. Unlike conventional neurotransmitters, which are stored in vesicles, this was the first atypical neurotransmitter to be identified. Because it's a gas, you cannot store it readily in vesicles, it can diffuse out. But instead, it is synthesized on demand when nitric oxide synthase binds to the target proteins. So subsequently, a lot of research has been conducted on the modes in which nitric oxide signals. So it is known to bind to the heme center of proteins and modulate the activity. It is also known to modify proteins by post-translational modification, which can regulate.
The work on the blood vessels, that also answered the question of how nitroglycerin relaxes blood vessels. And it had been known for a long time that like people with coronary artery problems where the arteries are getting clogged and the blood supply is reduced and they get chest pain, that nitroglycerin, they put it under their tongue and then their chest pain goes away. And the reason is it relaxes, it's called a nitric oxide donor. It essentially generates nitric oxide, which then relaxes the blood vessel, increasing the flow to the heart. And that probably does similar things in the brain.
Yes.
And that work also led to the, you mentioned this enzyme guanyl cyclase that's activated by nitric oxide to produce cyclic GMP. So that led to the development of drugs that increase cyclic GMP levels in cells. And everybody's heard about Viagra, and which is used for erectile dysfunction. But actually, there's also evidence now it can have some good effects on the brain, which is really interesting. There's a lot of interest in seeing whether it might benefit people with cognitive impairment. So nitric oxide then, so it's interesting. The way that nitric oxide is produced is that when cells are active, when they're stimulated, calcium moves into the cell. And calcium, through interacting with a protein calmodulin and then activating an enzyme, will stimulate this nitric oxide synthase to produce nitric oxide. So these neurons that are capable of producing nitric oxide, the main stimulus is activity in the neurons. And that led to the thinking, or contributed to the thinking, that maybe there's a role for nitric oxide in things like learning and memory, which is known to require neural activation, calcium influx, and so on. Yeah, and then you mentioned, so this is a gas, and normally you think of a neurotransmitter as being packaged in these vesicles in the terminal of an axon, presynaptic terminal, and then the neurotransmitter is released, and then binds to a receptor right there in the synapse. But nitric oxide is a gas, so it diffuses. It's not, and the evidence now is it's not just acting on one synapse, that the nitric oxide diffuses and may affect multiple synapses, which is kind of fascinating. So it may have a broader effect on the circuitry that's involved in things like learning and memory.
Exactly. And neuronal nitric oxide is also localized to the membrane of the, the nerve terminals, the synapses, where it interacts with the PSD95 protein, which is part of the synaptic density, and the NMDA receptors. So there is a coupling of all those, and it interacts with a protein called K-bond, which helps it like an adapter protein. So you can produce nitric oxide in close proximity to where it's needed, because all these small gaseous molecules can be intercepted on the way, because the cell is full of different proteins and ions and all kinds of macromolecules.
Yeah.
So you really need to produce them at the site where it's needed for the specificity.
And also, I had a scientist in my lab, Awu Cheng, who, this was a while ago, like 15 or more years ago, she had evidence that nitric oxide plays a role very early in brain development in promoting the, what we call differentiation of neurons from stem cells. So that as the stem cells stop dividing and the neurons start to grow axons and dendrites, then the nitric oxide is being produced. And now investigators study nitric. So the question is nitric oxide produced in the brain? The answer is yes. It can actually be measured through certain means of measuring it. The enzymes are there to produce it. But then the question is, how do you establish a role for nitric oxide in something like learning and memory or a disease? And to do that, you have to be able to either increase or decrease the nitric oxide production.
Yes,
right?
Correct. Yes, that's right. So our lab has got mouse models which lack the different isoforms of nitric oxide. So that would be an excellent tool to definitively show that the nitric oxide, because these are dedicated enzymes which produce nitric oxide, right? So if you delete them, you will be able to decipher their effects. So in some of these mice, you will find some learning and memory deficits also when these enzymes are knocked out.
Yeah.
But then I should also mention that these enzymes are not the only ones which produce nitric oxide. So nitric oxide is also produced in the oral cavity in our mouths, so from dietary nitrates.
Huh.
So the bacteria which reside there, they have nitrate reductases, which convert nitrate to nitrite. And then when you swallow your saliva, this gets, it goes to your stomach where the pH is acidic, and then that can lead to the production of nitric oxide. So that's why this is a new area of research, not so new, but it is catching up. Why your green leafy vegetables are good for your blood pressure regulation, because these leafy vegetables are a rich source of nitrates.
Huh.
So,
And if does that, so it's in the saliva. So if you chew your food more, if you chew these things a lot before you swallow them, will that enhance the?
Well, it should, because
it should, right?
Yes, the residence time is higher. So, and interestingly, many of these mouthwashes that we use, right, to clean our mouths, they have, they get rid of these salivary bacteria.
Oh.
So
yeah.
There have been reports in literature that after using mouthwash, your blood pressure changes a little bit if you keep using them, because you don't produce that much NO. So this is a very interesting field of research, which is both, being studied both in the hydrogen sulfide field as well as the nitric oxide field, that the nitric oxide, which is produced endogenously. A part of it comes from the mammalian cells. The other part comes from the bacteria, which grow in your body. So that is an interesting factor. And yeah.
But can you, so the nitric oxide, from eating that, the nitric oxide gas actually gets into the bloodstream and diffuses, or?
It may be, yeah. So there have been reports, I think Jonathan Stamler has also shown that it binds to hemoglobin in the blood.
Oh, I see.
And that is one way by which the nitrosylated hemoglobin, it can be transported to other parts of the body. So
and maybe the brain too.
Yeah, maybe the brain too, and cross the blood-brain barrier. There's a phenomenon called transnitrosylation where the NO is transferred from one protein to another. So there is a relay. So that could be one way by which the nitric oxide can be transferred.
So there's three ways we've talked about to increase nitric oxide levels: the eating green leafy vegetables, the drugs like Viagra that inhibit or increase the cyclic GMP levels, and then, well, nitroglycerin, or even, I was looking at some studies where there's certain medical conditions where they have the individual inhale nitric oxide gas and it's beneficial. There's something with like this problem with newborn babies. I was watching this video by Luis Ignario.
Hello, Ignaro. He has a new book on called Dr. No.
Yeah. Dr. No. Yes.
Yes.
That's a James Bond movie.
Yes.
Yeah. Okay. So the point there is that there's a lot of research being done on the therapeutic potential of manipulating nitric oxide levels using various means. I should also point out that exercise, physical exercise, will increase nitric oxide production.
Yes.
Probably also in the brain. And of course, physical exercise, if you do it regularly, will lower your blood pressure. So there's probably some role for nitric oxide in that. Okay, so let's briefly talk about carbon monoxide before we jump into your favorite gas.
Yes.
So carbon monoxide, how is that? So we know it's produced when things burn.
Yeah.
Right. But how is it produced in cells?
Okay, so here carbon monoxide, like nitric oxide and hydrogen sulfide, are also generated by enzymes. So, and this carbon monoxide is produced as a byproduct of heme metabolism. So the first place where carbon monoxide production was identified in vivo was in the spleen and the liver. So where you have the, the old red cells which are degraded. So the hemoglobin heme is being degraded by the enzymes called hemoxygenases.
Okay.
So they act on heme to degrade it to biliverdin, carbon monoxide, and ferrocyan. So hemoxygenases are the ones which produce carbon monoxide in the body. And that also produces biliverdin, which is acted on by another enzyme called biliverdin reductase to produce bilirubin. So CO is actually part of the heme metabolism pathway, and it also activates guanyl cyclase. So this is yet another gas, which we all know is very toxic because its affinity for hemoglobin is very high, and then it's known as a silent killer, right? Because it's a colorless, odorless gas, and if you don't have good carbon monoxide detectors and you close all the windows and you burn stuff or you have some electrical equipment which can generate carbon monoxide, that will lead to a very quick death. And that's what happens you see in the news often, like some couple who've gone to the different resorts, and all of those are attributed to carbon monoxide poisoning. But then we really need carbon monoxide for some of the endogenous functions.
So there's this heme oxygenase and the degrading heme in the red blood cells. Do all cells have heme oxygenase?
Yes, pretty much all cells have heme oxygenase, but there are different isoforms of heme oxygenase. So there are 3 isoforms like heme oxygenase 1, heme oxygenase 2, and heme oxygenase 3. So the heme oxygenase 2 is a neuronal one, but it's also present in the smooth muscles. And heme oxygenase 1 is the inducible enzyme, like it gets induced in response to all kinds of stress, oxidative stress, nitrosative stress, or anything. So it is. So there are different isoforms to take care of heme production or even biliverdin production in different cell types.
And what does the carbon monoxide do in cells after it's produced?
So it acts in different ways. So right now, one of the functions I mentioned is that it activates guanyl cyclase to produce cyclic GMP, so which is a signaling molecule, and cyclic GMP can modulate several signal transduction cascades, and that is one of the effects. And it's also involved in the vasorelaxation, which in different tissues, and it is present in the enteric nervous system, the gut. And it is present in the myenteric plexus, that region, and it is involved in the relaxation of the gut movements. So in some of these mice models, this there are GI tract disturbances as well, and a decrease in cyclic GMP production.
So it's acting essentially in the same way that nitric oxide is, at least from the standpoint of this cyclic GMP
yes
production. And so that seems kind of redundant.
Yes, that seems kind of redundant, but at the same time, this plays very important roles, during hypoxia or different conditions where some of these enzymes are active. So there is an interesting connection between hydrogen sulfide, which I'll come to later, and heme oxygenase. So it maintains the vascular tone in the brain. So all these heme oxygenases are oxygen-dependent enzymes. So during hypoxia, they are not activated. So it requires oxygen for the activity. So during normoxia, normal oxygen tension, so you get carbon monoxide, which is produced, which actually binds to the heme center of one of the enzymes which produce hydrogen sulfide and suppresses its activity. That kind of in normoxia, because you don't want that enzyme to be active during normal conditions. And during hypoxia, what happens is that the heme oxygenase does not have oxygen so that to generate carbon monoxide. So then the tonic inhibition on CBS, the enzyme, the heme enzyme, which produces hydrogen sulfide, is relieved. And so it's able to produce hydrogen sulfide, which plays very important roles in the mitochondria during hypoxia as well. So there are a lot of interconnections. So the redundancy is built in because at different conditions, the different enzymes get activated. So there is also spatial compartmentalization. Not all neurons which produce nitric oxide produce carbon monoxide. So that's why, although they all produce cyclic GMP, you take these enzymes and activates guanyl cyclase. You put them in a test tube, they are going to do exactly what the biochemical reaction tells you to do. But in the real world in cells, the scenario is very different. So,
yeah, that makes sense. Okay, now let's talk about hydrogen sulfide. Why did you get so interested in hydrogen sulfide?
Yes, so I got interested in hydrogen sulfide for multiple reasons. So one, at the time when I was in Saul's lab, I was working on sulfur-containing molecules. And this was, I noticed that Saul had these mouse models of hydrogen sulfide deficiency. Like there are 2 mouse models currently in a lab, which don't produce hydrogen sulfide like normal mice. And then at the time I was working on Huntington's disease, and I found that the CSE knockout mice, the CSE is enzyme which generates hydrogen sulfide in neurons. These mice had a phenotype which was very similar to that of Huntington's disease. Like they had a clasping phenotype. When you pick up a mouse which cannot produce hydrogen sulfide by the tail, it adopts a limb clasping phenotype. It adapts a bat-like posture. So it starts clasping its hind limbs. And this looked very similar to the Huntington disease mouse model. And one of the features of the Huntington disease mouse model is, the motor deficits and some cognitive dysfunction, and also oxidative stress, which is something which I study a lot. And these exact same phenotypes were seen in the CSA knockouts. So I thought, why not look at hydrogen sulfide and its role in all these neurodegenerative diseases. So that was the starting point. It was a simple observation, like the Huntington mice look very similar to the CSE knockout mice. So there must be some connection. So
and those mice that aren't producing the hydrogen sulfide normally and have these problems controlling their body movement, do neurons die in their brains like they do in Huntington's disease?
So what I have observed is as a function of age, there seems to be a shrinkage of the striatum even in the CSE knockout mice, although it's not as pronounced as that in Huntington's disease, where the striatum atrophies to a very significant extent in that the ventricles look very enlarged. But in this case, you do see some of those morphological changes. So there is some similarity, but not to that extent.
And so I think the first time, if I remember, that I learned about hydrogen sulfide and it can have dramatic effects, there was this very provocative paper in Science in, well, I don't remember. But essentially it said that if they give mice hydrogen sulfide gas, it puts them in a state of suspended animation where their body temperature goes down to like room temperature, and so they're very abnormally cold, but they stay alive, and like they can keep them in that state a long time, and then they remove the hydrogen sulfide and they recover and seem healthy. And I was thinking, and then people kind of jumped on this and said, oh, maybe on space trips where the astronauts have to travel for 200 years to get, I don't know, to some other planet that's likely out there that has life, that they can breathe this hydrogen sulfide and they'll be in this suspended animation where they're not really aging much, and
right.
Kind of science fiction. But so I guess my point is it has like dramatic effects on metabolism. So I'm sure you saw that paper too, right?
Yes, it is a 2005 paper by Mark Roth from Mark Roth's group, where they show that in small rodents, especially the mice, if you expose them to about 80 parts per million of hydrogen sulfide, they go into the state of torpor and suspended animation. And this occurs when the temperature is also low, like 15 degrees or so. And then later on, when you elevate the temperature and also get rid of the hydrogen sulfide in the room, or you bring them to a normal atmosphere, then this effect goes away. So this has been primarily attributed to the inhibitory effect of hydrogen sulfide on the mitochondrial respiration. So hydrogen sulfide at high concentrations can inhibit the complex, the cytochrome C oxidase, which is part of the electron transport relay, which generates ATP from oxygen, right? The very last step. So that step is affected by hydrogen sulfide. And so this is what causes, because the oxygen consumption rate of the organism that drops and less ATP is produced. So everything, all the metabolic activities are suspended. So
and there may be fewer free radicals produced too, because mitochondria.
Yes and no. Also, yeah, there would be some free radicals produced also, but again, it depends on the direction of the electron transport. It is more complicated than mentioned. But yes, but this is primarily the reason why we have this suspended animation state. However, other investigators have tried to replicate it. It doesn't work very well for like large organisms. But right now it is being considered as in the medical field, like before ischemia reperfusion, or you just put the, especially people on ventilators, or it can prevent ventilator-induced injury. So those are the aspects that are being currently explored. And also before a major surgery, if you can administer hydrogen sulfide, the recovery will be better. So,
and also may have an anti-aging effect, right?
And
so, all our work on intermittent fasting and caloric restriction, and
exactly.
Now I think those, so there's a number of labs that provided evidence that caloric restriction and fasting can elevate hydrogen sulfide, and that may mediate some of the health benefits of, reducing energy intake.
Correct, that's right. So the paper that you're referring to is from the late J. Mitchell's group. It was a Cell paper in 2015, where they show the benefits of dietary restriction is mediated by hydrogen sulfide.
Yeah.
So, and also they use the same model system to show that during ischemia reperfusion, before that, if you put the mice on, dietary restriction, or you administer H2S, that actually makes the recovery better. So it fits in more ways than one. Many times when you have a major surgery, they tell you not to eat a lot.
Yeah.
So that's one of the beneficial effects, which I don't think the doctors know right now, is that hydrogen sulfide is also produced. So this is a very interesting field also. And the other component, which you said, the exercise. And there have been some reports out there in literature that if you put mice on a treadmill, then the production of hydrogen sulfide is increased. So I won't be surprised, like dietary restriction, exercise, and both of them being anti-aging, of course. And they also end up producing hydrogen sulfide. So.
Okay, and so now you mentioned this, did you mention it? The CSE knockout mice. So mice that can produce the hydrogen sulfide, they have the phenotype similar to Huntington's disease. So I guess, so Sal and you are really good at the biochemistry now. And can you explain how hydrogen sulfide affects the function of cells? Because it's not the same as nitric oxide and carbon monoxide, which increase the cyclic GMPs. So can you take a little time and kind of walk us through that?
Yes, sure. So first of all, to understand this, how hydrogen sulfide acts, we have to go to the pathway through which hydrogen sulfide is produced. So in the body, we have a pathway called the transsulfuration pathway. So through which hydrogen sulfide is produced. In fact, hydrogen sulfide is derived from the amino acid cysteine, which in turn is derived from dietary methionine. So to keep it simple, there are three enzymes which produce hydrogen sulfide. Cysteine gamma-lyase, CSE, which is the major enzyme present in neurons. Then you have cysteine beta synthase, or CBS, which is present in astrocytes. And then you have a third hydrogen sulfide producing enzyme called 3-mercaptopyruvate sulfur transferase, which is present both in neurons and astrocytes, but it's also present in the mitochondria. But then 3-mercaptopyruvate sulfur transferase acts on 3-mercaptopyruvate to produce hydrogen sulfide, whereas the other two use cysteine as a substrate to produce hydrogen sulfide. And so once again, you come back to the same question, right? Why do we have 3 enzymes to produce one gas? The reason is they are, segregated spatially. So CSE is the neuronal enzyme and CVS is the astrocytic enzyme, and 3-MST acts under different conditions. So now once these enzymes produce hydrogen sulfide can act in multiple ways, a lot of ways to modulate the activity. Almost all cellular processes are regulated by hydrogen sulfide. So the first thing is regulation of blood pressure, which was the first demonstration from the Snyder lab, showing that it's important for vasorelaxation. It also stimulates guanyl cyclase, but in addition, hydrogen sulfide inhibits phosphodiesterase, which degrades the cyclic GMP. So it acts in 2 ways to boost the cyclic GMP levels. But that is the vascular function, of hydrogen sulfide. But hydrogen sulfide also has a lot of other functions in the nervous system. And one of the ways by which it modulates this is by a post-translational modification termed sulfhydration, which is analogous to nitrosylation. So hydrogen sulfide can convert the SH groups of cysteine residues to an SSH or persulfide group. And in doing so, it can modify the activity of the protein concerned. So
so you mentioned the amino acid cysteine,
yes.
Right? And so the hydrogen sulfide, which is H2S, 2 hydrogens, 1 sulfur, yes, that's interacting with the cysteine, and then you get like cysteine has sulfur in it, right?
Correct.
So you're getting bonding between the sulfur on the cysteine with the sulfur on the hydrogen sulfide?
Yes. So hydrogen sulfide, this chemistry is a little bit more tricky because hydrogen sulfide cannot directly modify, but in the end, you get like a disulfide, persulfide bond. So you get SSH.
Okay.
So you get like a disulfide bond kind of formation, to explain in simple terms.
So that action of the hydrogen sulfide through this process on the cysteine. So then the question becomes, does hydrogen sulfide only affect the function of proteins with cysteines? I imagine a lot of proteins have cysteines.
Yes.
Is that the only way it's affecting the proteins, by interacting with the cysteines?
No, there are other methods. This is one of the very common ways, but although you have a big proteome, only 2%, cysteine comprises only 2% of the proteome, and it's mostly present in some active site or very important regions. So by modifying that, it can change the activity. But hydrogen sulfide can also bind to heme centers of proteins, just like the way nitric oxide and carbon monoxide does that. Okay. So it can convert the ferric to ferrous and then modulate the activity that way also. And that's what it does with the guanyl cyclase. It's a heme enzyme, right? Heme containing enzyme. So it's very interesting that hydrogen sulfide can act in this way. So a third way by which hydrogen sulfide acts is, it is the only inorganic molecule which can donate electrons to the electron transport chain. So, and those electrons can be relayed. And this is operational during conditions of hypoxia also, especially because when you have normal oxygen, hydrogen sulfide can get oxidized. But, and so that's what happens. So hydrogen sulfide takes over. It's a backup mechanism. Like in the moment when you're undergoing hypoxia, you need some systems to take over so you can maintain life before the paramedics or medical staff arrive. So hydrogen sulfide does precisely that. It takes over because it's no longer degraded by the mitochondria. by the sulfide oxidation unit. So it can donate electrons and then maintain the flow of electrons in the electron transport chain and produce ATP. So, and that will sustain life. So this is actually a backup mechanism during conditions of hypoxia. So H2S can act in multiple ways, by binding to heme centers, by modifying proteins, and also by donating electrons to the electron transport chain.
Now, when you mention ferrous iron, I think of free radicals production. And it turns out that iron can be toxic to cells, and it kind of facilitates the production of a particularly damaging free radical, hydroxyl.
Yes.
Now,
yes.
And then now also, I don't want to jump around too much, but now I'm thinking of nitric oxide, and we failed to mention that nitric oxide is a free radical.
Yes,
right?
It is a free radical.
And that nitric oxide, if it gets too high levels and isn't removed, it can actually lead to free radical damage to proteins and other cellular constituents, which can be bad. So there seems to be this fine line between healthy, optimal levels of nitric oxide, carbon monoxide, hydrogen sulfide, and toxic. And there's now quite a bit of evidence that, for example, in Alzheimer's disease, there may be excessive nitric oxide mediated damage to neurons. You want to talk about kind of the dark side of the gases?
Oh yeah, so that's right. So all these gases have a dark side. Carbon monoxide, you already know that if you have a little bit extra, you can die. And with nitric oxide, if you have high levels of nitric oxide, which Mark just mentioned as a free radical, it can react with superoxide, which is another kind of free radical, which is produced by the mitochondria, and it forms the highly reactive peroxynitrite, which can, modify proteins again and cause a lot of nitrosative damage in cells. So that is the dark side of nitric oxide. Like if you have excess, this is what is going to happen. It's going to cause nitrosative stress. And Jonathan Stamler is an authority in this field of nitrosative stress. So he would be able to, really talk about this aspect. But then, of course, because I'm in the field of redox, this peroxynitrite is a major culprit in damaging all the proteins and lipids and all the modifications which occur on them. And that could lead to accumulation of damage, which can, depending on the disease. In Alzheimer's disease, you have elevated nitrosative stress. We see that in Parkinson's disease, there is excess nitrosylation because there is excess NO being produced and decreased sulfhydration. So that is a feature that we have been observing. But of course, we really need nitric oxide also for normal functioning. But if it crosses a certain threshold, it's going to cause all these kind of damage. You know that very well. If you have, we need oxygen for life, but we have too much oxygen, that's a problem. So that's where the field is going to. Even the same thing with hydrogen sulfide. You have excess hydrogen sulfide, it can inhibit the mitochondria and then shut down life. So the goal is to now induce low levels of these so-called toxic molecules. So it, will adapt the organism to respond to stress better. So that, Mark, more than anybody else, the concept of hormesis, right? So that's what is.
I'm going to have a podcast on hormesis, that, what doesn't kill you makes you stronger. Notion. And there's a lot of evidence for that kind of mild stress or challenges with, so exercising your neurons increases production of all of these gases. And they're potentially toxic, but there's these adaptive mechanisms. We don't have time to get into this in this podcast, but so, for example, some of these gases actually increase they stimulate genes that encode antioxidant enzymes.
Correct. Yes.
that remove the free radicals.
Yes.
You know, so when your cells are active, there's more free radicals produced, but it's okay because there's these evolutionarily mechanisms, ancient mechanisms that are activated that actually make the cells more resistant to even higher levels of the stressor. Yeah.
Yeah, that's true. Because even if you look at NMDA receptor signaling, you have excess NMDA activation, you have too much of all these molecules produced. So I've always noticed that in biological systems, some of these stresses which induce damage at higher doses also trigger a counter response.
Yeah.
So in this case, like you mentioned, for example, if you have hydrogen sulfide, it triggers the NRF2 signaling pathway, which is like the master regulator for all antioxidant defense. So any of these processes, if you think it's going to cause damage, it is also inducing the corrective mechanism at the same time. And when the corrective mechanism fails to restore the balance, that's when the balance tilts over to the
this
bad
is
side.
a major problem with people who are sedentary and overeat. Their cells, essentially, they're not being challenged. And so they become complacent.
Yeah.
And their defense systems to protect them against a variety of stressors are downregulated.
That is true.
Yes,
in fact, in obese people, it has already been noticed that hydrogen sulfide levels are lower. So even if you do a short walk, you don't have to be a marathon runner. Even if you just walk for 15 minutes a day, it will go a long way in preserving your health. Oh, this is just like climbing a mountain, say Mount Everest. You don't straight away climb all the way to the top. You have all these base camps. Why do you have it? Because you have to adapt. So that the first climb is your actual stress, which will adapt you, so that you're in better shape and you produce more hemoglobin or, oxygen carrying molecules. And then you go on to climb. So vaccination is the other adaptive response, right? We know that, especially we know that the SARS virus or any of the antigens, they are damaging, but then you do inject a small amount so you can build a response. So there are different kinds so that you can deal with a higher dose later on. So this is the same thing for gases. You need to maintain them at a basal level. And when you are being stressed, like say exercise or running uphill for climbing stairs, you'll produce a little bit more of it, but it will be consumed.
Yeah, and then in a lot of in some murder mysteries, people who want to kill somebody, they'll get themselves adapted to high levels of, say, arsenic, which is a toxic chemical. They'll take a little and a little more, and then they'll sit down and whatever, drink or eat food that has arsenic with the person they're trying to kill.
Yeah.
And the person, that they're trying to kill will die because they're not adapted to the toxic.
Yeah, it is the same thing.
Oh, is that true for carbon monoxide too?
I'm not 100% sure about that, but I think because of the affinity for whatever you do, you cannot bypass the affinity of carbon monoxide to hemoglobin. So it will kill you. You can't bypass that.
Yeah, that's true.
So it wouldn't work there.
Okay, what's your latest work with hydrogen sulfide, and what are your future directions?
Okay, so right now we are trying to elucidate the role of hydrogen sulfide in Alzheimer's disease. We have already shown that when you administer molecules which generate hydrogen sulfide, the mice get better, their learning and memory is improved. So now we are going ahead.
What molecules are you giving them?
So there are different types of hydrogen sulfide donors. One which is a general donor, and a slow release donor which donates hydrogen sulfide at a low rate. Like it's called GYY4137, and a second one is a fast release hydrogen sulfide donor called NaHS, sodium hydrogen sulfide. And A third type is you can target it specifically to the mitochondria. So we are evaluating a series of these compounds, how much to give and what dose to give. And we are trying to look at the proteins which are modified in Alzheimer's disease. So we already know that if you take the brain, look at the brains of patients with Alzheimer's disease, they have lower levels of hydrogen sulfide modified proteins, lower levels of sulfide-rated proteins, and their learning and memory is really compromised. But then when you give these mouse models of Alzheimer's disease, which have compromised learning, you give hydrogen sulfide, they get better. So that is the area that we are getting into. And I'm also trying to develop compounds which can stimulate the transsulfuration pathway to produce more hydrogen sulfide. So hopefully we'll come up with molecules which can ramp up this pathway, and that can be used. And some of them can be natural too. So those are the areas that we are currently looking at.
That's exciting from the standpoint of potential clinical applications in patients, right? And have any of these molecules you're using in the mice to increase hydrogen sulfide levels been given to humans?
Not the ones which I'm working on right now, because we need substantial funding to get into, but there are other groups like John Wallace's group from Canada, Antibody Therapeutics. There are some H2S donors like ATB346, which are in clinical trials right now, but not for neurodegeneration, but for different conditions like as an anti-inflammatory, non-steroidal anti-inflammatory drug. So there are some of those molecules which are under trial. But then one interesting thing which I must add is that garlic, it's known to lower blood pressure. It acts through hydrogen sulfide because it has certain sulfur containing molecules which can generate hydrogen sulfide. So that's another. There's a PNAS paper out there which shows that the vasoactivity of garlic can be attributed to hydrogen sulfide.
And probably onions too.
Yes. The allicin, yes, all of those. Broccoli, sprouts, and many of those have sulfur-containing molecules.
Yeah, I'd like to eat. I used to eat a lot of garlic, but so I've been having some problems with gastric reflux. So that's, the spicy foods are causing problems. So, okay. But, go ahead.
At the end of the day, we might oversimplify it and saying that my grandmother was right: eat your greens, exercise, and sleep well, and then you will. But then the mechanism, we are trying to unravel the mechanism.
Yeah.
Which this is ancient wisdom, but then we are finding out the roles for all these molecules.
Now you just started in your faculty position, tenure track faculty position at Johns Hopkins. How are you coming along and starting to build your lab up? That's always, for me, I think for most scientists, it's really exciting, the most exciting time probably.
Yes, it's the most exciting time, and things are going on really well. So I just secured an R01 and an R21 grant. As well as
congratulations.
Thanks. And also a grant from the American Heart Association.
Oh.
So things are going well. And, I've already hired 3 postdocs in the lab right now.
Wow.
So we are going to be looking at all the neurological diseases and basically delve deeper into the gasotransmitter field. So we're really excited about it, setting up the lab, buying new equipment, and the Department of Pharmacology is a very nice setting where, it's very famous for its mass spectrometry, which we require for these kind of studies to look at post-run. So yes, things are going well. And I'm also going to organize a conference in 2024 on hydrogen sulfide in Washington, DC.
Oh, so, great.
Yeah.
Huh. In what year?
2024.
Okay, that's coming up pretty quickly. And
yes.
And COVID's over, so it can be in person.
Yes, the last one was held just a month back in May in Budapest, and that was an in-person meeting too. So
so this is what you're saying is it's really remarkable how quickly this hydrogen sulfide field has moved forward. Now there are probably hundreds, maybe thousands of scientists throughout the world working on it.
I would say it's still a pretty small group, but more and more people are interested in the field because of its, very broad implications. You can apply it not just in neuroscience, but, also in the immune system or in the cancer biology where it is, and interest in diseases like, COPD. You can even chronic fatigue. That's another project that I have.
Oh.
So hydrogen sulfide is dysregulated in chronic fatigue as well. And due to the pandemic, like COVID-19, many of the people who recover from COVID-19 go through fatigue for lasting more than 6 months. So now they are classified as chronic fatigue patients, and their plasma levels seem to show a difference in the levels of hydrogen sulfide and nitric oxide. So that's an area of intense investigation as well.
Are there any animal models of chronic fatigue?
No, not really. We don't have any animal models because the closest they even get to is to make a mouse swim for like 6 hours or something like that.
Oh.
So it's not really a real model, but I suppose, most of these diseases like chronic fatigue and COVID, they have this inflammation associated with it.
Yeah.
And so you could probably inject all the cocktail of cytokines and make the mice really sick. And so
yeah, or there's this bacterial molecule, LPS.
Yes.
Lipopolysaccharide.
Yes.
Okay.
Yes. So there are no real mouse models. So.
All right. So you think there's a bright future for translating the basic research into helping people improve their health? Yeah.
Yes, definitely. I'm 100% sure about that. We just have to find the right timing and the right molecule to do it with.
Now, yeah, it's not really practical for people to be inhaling hydrogen sulfide gas,
no.
Right? Because it's kind of tricky business, and yeah. So
yes, you can't inhale. So probably a pill or something which will, release hydrogen sulfide, or it can be an activator, which say crosses the blood-brain barrier and activates. There are, many drugs which people explore as pills, especially in Huntington's disease.
So there are drugs that activate the heme oxygenase. Oh, no, sorry. That's that.
I'm mixed up. Yeah,
CSE, yeah.
Yeah, so there are some small molecules which can upregulate H2S production. For example, I do know that the anti-aging drug metformin, it induces the production of hydrogen sulfide.
Oh, interesting.
Yeah, so many of these, it's already reported in literature. So some of these pills, they induce the production of hydrogen sulfide. There have been reports that resveratrol, the component in wine, that increases H2S production. So I'm pretty sure we have to find which of these is the most potent one.
Yeah.
So they act through multiple ways. So any of these you can consume, you don't have to inhale a gas, you can just eat food or drink wine, but okay. It all depends. So I'm not surprised why the Mediterranean diet is also very beneficial. A lot of those foodstuffs which they consume may generate hydrogen sulfide, and so you don't have to see it, but it at least prevents cognitive decline.
And so early on, we talked about bacteria producing hydrogen sulfide. Are there certain species of bacteria in the gut that produce hydrogen sulfide?
Yeah, there are several species of bacteria. Even E. coli produces hydrogen sulfide, but but there are some specific sulfate-reducing bacteria, and it's a whole field. Like, I'm not an expert on that area, but there are different species which produce hydrogen sulfide, and right now, there are scientists looking at what is the contribution of the gases produced by the microflora in your intestine to the well-being. You could have bacterial metabolites which enhance cognition. And so, yeah, and in fact, some of the, I think, B vitamins are also produced by the bacteria, which you absolutely need as a cofactor. So I'm pretty sure that some of these molecules are, they maintain the colon health. In fact, there is an adaptation which takes place in mammals to counteract this, because there's a lot of course, H2S is a part of flatus, right? So so a lot of these bacteria in the colon produce so much hydrogen sulfide. The cells lining the colon have to have a very active sulfide oxidation unit in the mitochondria to get rid of, detoxify this gas.
So
that is another area which people work on. So there are scientists working in this field also.
Because it's interesting that so we talked about obesity being bad and less hydrogen sulfide and caloric restriction, increasing hydrogen sulfide. And so it's been shown that people with obesity have an unhealthy gut bacterial composition. And there's even evidence, mostly from animals, but I think some early human studies, that if one does fecal transplant and puts healthy bacteria in the gut of animals or humans with obesity, it can help them get their weight down. So yeah, there could be an important role for hydrogen sulfide in the beneficial, through the gut, in the beneficial effects of caloric restriction, maybe even exercise.
Yes, that's true. Exercise is certainly very interesting. So you should really talk to Jonathan Stamler about exercise and so.
All right, we covered a lot of ground, and you really highlighted the importance of nitric oxide, carbon monoxide, hydrogen sulfide in normal brain function, things like learning and memory, and possible deficiencies or abnormalities in production of these gases in neurological disorders, and then the potential for development of treatments, including drugs, that can kind of optimize levels of these gases in the brain. And so it's very exciting research. And keep up the good work, Bindu. I've enjoyed talking with you.
Yeah, thanks, Mark. Yeah, I think we should continue the conversation. It's really a blossoming field.
Yeah, we can talk. Yeah, I'd like to talk with you offline if you want to about get into the more of the science, because we don't really get very deep into at the molecular level or.
Yeah, there's a lot. There's a lot. Yeah.
Okay, Bindu.
Okay, bye.
Thanks. Bye.